Hello, everyone. I am Arxel G. Elnar, a postdoctoral researcher under the BK21 program of the Department of Animal Science and Technology, Chung-Ang University. Today, I will be sharing a presentation which is a part of my doctoral dissertation entitled Isomer-specific CLA bioconversion of Bifidobacterium breve, a comprehensive study on linoleic acid isomerase. The contents of this presentation will include an introduction, followed by an intergenera comparison of linoleic acid isomerase, before focusing on the bifidobacterial linoleic acid isomerase, followed by the characterization of its enzymatic activity and its potential for industrial applications, to be concluded with a summary and future perspective part. Moving on with the introduction, I think many of us will be familiar with the term probiotics. So probiotics, by definition, are live microorganisms that, when administered in adequate amounts, would confer a health benefit on the host. Now, similarly, postbiotics, although a relatively new term, can also elicit similar health benefits on the host. However, they are composed of inactivated cells or cellular components. So the main difference between pro and postbiotics lies on the viability of the cells present in these preparations. Now, the general scheme of probiotic studies would actually begin with the identification and characterization of the potential probiotic candidates, as well as its application, in order to prove that they can improve the health of the host. Now, many of these probiotic candidates can actually be divided into two broad categories, namely traditional probiotics, which includes the genus that many of us would be familiar of. So they include Lactobacillus, Bifidobacterium, Propionibacterium, Saccharomyces, and Streptococcus. And the next generation probiotics, or NGPs, which includes Eubacterium, Akkermansia, Faecalibacterium, Roseburia, and Bacteroides. Now, in this presentation, we will be focusing on Bifidobacteria as probiotics. The name Bifidobacterium comes from two Latin words, bifidus and bacterium. So bifidus means cleft, split, or divided, and bacterium means small rods. So we can see the etymology of this name comes from the observation of its cellular morphology, having irregular or branch shape. Now, here are some of the textbook definitions for Bifidobacteria. So they are considered high GC, gram-positive, and anaerobe or microaerophilic bacteria, and they are considered to be widely accepted as traditional probiotics with numerous health benefits, as well as part of the normal microbiome of the human GIT, given that they are also one of the first colonizers of the human infant gastrointestinal tract. Now, in this figure, we can see the proposed health benefits of using Bifidobacterium as probiotics. So in our study, we use the strain JKL 2022. So Bifidobacterium breve JKL 2022 has been shown to be a strong CLA producer. Now, on top of its inherent probiotic properties, the strong CLA production is actually seen to improve some of its health benefits, including anti-obesity, anti-diabetes, anti-cardiovascular disease, anti-cancer, anti-tumor, immunomodulation, lipid metabolism, and promotion of the eubiosis and disease prevention. Now, in this slide, we can see a graphical summary of the potential modes of action behind the benefits of Bifidobacterium on gut health. So it starts with the colonization and establishment of the niche of Bifidobacterium, which will be followed by the cross-feeding with butyrate producers. Now, the butyrate produced can actually improve the intestinal barrier through the enhanced expression of tight junction-related genes and proteins and increased mucin expression. In addition, they can also do competitive exclusion of pathogen attachment through the production of antimicrobial substances, including bacteriocins and short-chain fatty acids. Lastly, they can also modulate the immune response of the host through several mechanisms. And in figure 4, we can see that the beneficial effects of common probiotic Bifidobacterium strains, and we can see that there are a lot of Bifidobacterium species currently being studied to determine their specific effects on host health. So this includes facilitating the nutrient absorption, anti-inflammation, reducing fat accumulation, promoting psychological health, regulating host immune system, as well as anti-cancer, anti-infection, antivirus effects, and also improving the bone health. So this puts Bifidobacterium as a key player in many of the gut organ axes. Now we move on to describing conjugated linoleic acid. So it is also known as rumenic acid. Now, conjugated linoleic acid or CLA is a generic term for the geometric and positional dienoic isomers of linoleic acid. So it is composed of 18 carbon with two double bonds, and the most abundant isomer would be the cis-9 trans 11 CLA. So the cis-9 trans 11 CLA has been proven to have numerous health benefits, including anti-obesity, anti-inflammatory, anti-cancer, immunomodulatory, and improvement of the cardiovascular health. So this coincides with the innate probiotic properties of Bifidobacterium. However, one concern remains, and that is the low abundance of CLA in food system. So in this slide, we can see the CLA content of various food items. So the highest coming from milk and dairy products, which ranges from 4.5 to 6 milligram per grams of fat, followed by meat, including pork and poultry and ruminants, fish and seafood, and vegetables. However, the vegetable oils can only be chemically processed to produce the CLA supplements that is available in the market. Conjugated linoleic acid is present in the market in the form of dietary supplements. The market price for conjugated linoleic acid ranges from 18.10 to 60.85 US dollar per kilogram in 2024. And based on the market statistics done by Grand View Research, it is expected to steadily increase until the year 2030. And dietary conjugated linoleic acid actually comes from vegetable oils through chemical synthesis. However, this process creates a mixture of different isomers, majority of which coming from the cis-9 trans 11 and the trans 10 cis-12 isomers. However, in our study, we were able to observe that Bifidobacterium breve can form specific isomers of conjugated linoleic acid, and studies which demonstrated the use of washed cells have comparable conversion rate compared to growing cells. In addition to that, we also observed that only Bifidobacterium breve JKL 2022 showed CLA conversion using washed cells and crude protein extracts. So we wanted to answer the following questions. First, is the CLA converting property of JKL 2022 a strain-specific property? Second, what are the underlying mechanisms that regulate this phenomenon? And third, what are the key enzyme or enzyme systems that catalyze the conversion of LA into CLA? So with that, we prepare different cell preparations of Bifidobacterium breve that can convert linoleic acid into CLA using growing cells or probiotic cells, inactive or washed cells or the postbiotic preparations, as well as the crude protein extract. So from there, we kind of built this project focusing on Bifidobacterium breve, focusing on the strain and enzyme characterization as well as probiotic development, leveraging on the microbial biosynthesis of linoleic acid into conjugated linoleic acid. Next, we will discuss the linoleic acid isomerase and intergeneral characterization. Animals can also produce conjugated linoleic acid, particularly ruminants. So through the activity of several microorganisms, which we refer to as microbial biohydrogenation, the starting linoleic acid and linolenic acid can undergo a cascade of reactions, forming conjugated linoleic acid and vaccinic acid. Now, these intermediate metabolites can migrate into the mammary tissue and undergo endogenous metabolization, in which the vaccinic acid, through the activity of the delta-9 desaturase, can be converted into conjugated linoleic acid. Now, the CLA formed in the mammary tissue can actually be incorporated into the host tissues or in the milk. However, when we look at the specific enzymatic pathways for the CLA bioconversion, it actually differs depending on the species of the bacteria. As we can see in Propionibacterium, it converts linoleic acid using the PAI enzyme or the polyunsaturated fatty acid isomerase primarily into the trans 10 cis-12 CLA isomer. Now, when we look at Lactobacillus, it actually uses a three-enzyme system composed of the MCRA, CLA dehydratase, and CLA decarboxylase, forming several intermediate metabolites and a combination of different CLA isomers. Now, for Bifidobacterium, however, it has long been hypothesized that there is a homologue of the MCRA enzyme that forms the rumenic acid or the well-known cis-9 trans-11 CLA isomer, which can be converted directly into the trans-9 trans-11 isomer. However, we later identified the key enzyme for Bifidobacterium, which I will discuss later. So several studies have already proved that Bifidobacterium breve is among the strongest CLA producer among different Bifidobacterium species, as we can see in this table. And when we take a step back and compare Bifidobacterium breve with other lactic acid or probiotic candidates, we can see that it is still one of the strong CLA producers. However, when we look at the catalysts used in these studies, most of them actually use growing cells or live probiotic cells, whereas only a number of studies actually use postbiotic preparations in the form of washed cells or crude enzymes. In our study, we were actually able to observe that Bifidobacterium breve, besides producing conjugated linoleic acid using their growing cells, they can also convert conjugated linoleic acid using postbiotic preparations, whereas other Bifidobacterium breve strains actually failed to produce the same reaction, which merits the question, why is it that only Bifidobacterium breve can produce CLA using its postbiotic preparations? So in our study, we were actually able to identify the gene encoding for the enzyme responsible for the CLA bioconversion of Bifidobacterium, and we then proceed with the checking for the LAI evolution and distribution analysis. So in this figure, we can see that Bifidobacterium breve JKL 2022 LAI enzyme is actually highly conserved among Bifidobacterium breve. So showing greater than 99% sequence homology among different Bifidobacterium breve strains. In addition, we were also able to observe that in other GIT-associated bacteria, that is non-Bifidobacteria, they also possess a significant LAI sequence. So this includes the genus Gordonibacter, Rholtibacter, Egerthella, Parulcinella, Collinsella, and Clostridium. So we can see that in this figure. Now, more than 100 sequences were used to create this dendrogram, and we can see that the LAI gene exhibits high degree of conservation among Bifidobacteria, which is higher than 95%, while a conserved domain was detected among non-Bifidobacteria gut-associated microbes. So in this part, I will be focusing on the Bifidobacterial linoleic acid isomerase, including how we were able to identify the gene encoding for the enzyme linoleic acid isomerase of Bifidobacterium breve JKL 2022. There are a lot of different lipid biosynthesis genes, and one of which, the reaction R03627 of butyric which is specific for the cis-trans isomerase, have already been annotated. However, when we look at this counterpart for Bifidobacterium breve, such as that for JCM7017 strain, it is not yet elucidated. So we leverage the existing whole genome data of Bifidobacterium breve and try to identify similar functioning proteins or enzymes present in their genome. However, most of the annotated proteins does not function for CLA biosynthesis. And so we moved on to studying the whole genome of Bifidobacterium breve JKL 2022, and the basic genome attributes are presented in table number 3. We then performed a whole genome comparison between different Bifidobacterium breve strains and found that there are no exclusive genes for JKL 2022 that would explain why JKL 2022 can convert CLA using its postbiotic preparations. However, we were able to determine that there are repeat copies of tyrosine recombinase XCRD. In addition to that, genes for lactobacilli CLA conversion was also found in JKL 2022 and other Bifidobacterium breve strains, specifically oleic hydratase or the MCRA, enolase, short-chain dehydrogenase, and a 3-oxoacyl carrier protein reductase. However, when we checked for the activity of these genes in vitro, they were also not functioning for CLA biosynthesis. And so, we investigated the proteome and transcriptome profile of Bifidobacterium breve JKL 2022. So in this approach, we tried to determine if there will be any difference in any of the proteins or transcripts when we expose the JKL 2022 in the substrate linoleic acid. So to do this, we basically grow JKL 2022 in a culture media with and without linoleic acid and perform total proteome and total transcriptome analysis. And from that, we were able to see that indeed one hypothetical protein, the JKL 2022-00014, which putatively encodes LAI or linoleic acid isomerase, is actually upregulated in the presence of linoleic acid. So this means that when the substrate is present, the gene encoding for LAI is actually expressed more. And from there, we did further investigation to confirm and validate if this is indeed the gene encoding for LAI. And to do that, we perform some protein-protein interaction analysis. So first, we perform a PPI analysis that predicted JKL 2022 LAI is distantly related to that of the Lactobacilli CLA biosynthesis genes. So this proves that Bifidobacterium actually utilizes a unique enzyme or enzyme system from that of Lactobacilli. We also performed a relative gene expression of CLA-related genes, and we observed that the LAI gene is much more strongly expressed in both JKL 2022 and 7017 as compared to the other CLA-related genes for Lactobacilli. And also, we were able to determine that JKL 2022 has a stronger basal expression of LAI compared to the JCM strains. So this was further validated in vitro, wherein we compared the postbiotic preparations of JKL 2022 and the JCM strains and observed that only JKL 2022 can convert linoleic acid into conjugated linoleic acid using its postbiotic preparations. So what it means is that even without the presence of LA, JKL 2022 strain can produce significant amounts of the enzyme responsible for the CLA biosynthesis, whereas the other Bifidobacterium breve strains failed to produce so. So in this case, the JCM strains are only upregulated only in the presence of LA. And from there, we went on and investigated why is it that JKL 2022 strain has a stronger basal expression of the LAI gene, whereas the other Bifidobacterium breve strains do not. So we tried to determine if there is any transcriptional regulatory mechanism responsible for this observation. And through several in silico analysis, we were able to predict that the TET-R and the LAI promoter region shares a strong interaction. So JKL 2022 genome actually harbors four different types of TET-R, but only two showed strong interaction with the promoter region of the LAI gene. And so we did a gene co-expression analysis using QRT-PCR and observed that LAI and TET-R217 actually share a similar pattern in the presence and absence of linoleic acid. So it means that there is a strong correlation between the expression of TET-R that would regulate the expression of linoleic acid isomerase enzyme of JKL 2022. So this was not present for the JCM strains. However, this is still an ongoing study, so there's still a lot to be confirmed and validated. But for now, at least we see that the ZR is strongly correlated with that of the expression of the LAI gene. And lastly, we performed some structural analysis and saw that the conserved LAI domain, which is also present in the non-Bifidobacteria genera, actually coincides with the predicted LA binding site. And through molecular docking and structural analysis of the JKL 2022 LAI, we were able to confirm the binding site of LA and LNA in LAI. Now, although we were able to identify the gene encoding for the LAI enzyme, there is still very limited literature regarding the characterization of Bifidobacterium breve linoleic acid isomerase. And here are some of the examples of the recent publications regarding the Bifidobacterium LAI. Now, this is still an ongoing study, and there is still a lot left to be discovered for the Bifidobacterium linoleic acid isomerase. We will now proceed with the enzymatic activity of bifidobacterial LAI. For probiotic studies, it is crucial that we validate the desired probiotic property that we want to observe in vitro. So to do that, we perform growth profile and CLA conversion of Bifidobacterium. So this assay used a control and a treatment group, the control without linoleic acid substrate, and the treatment with LA exposure. So when we look at the control group, we can see that the pH and growth curve is actually pretty standard. During the course of its incubation, the pH drops from approximately 6 to around 4.5, which suggests proper fermentation of the sugars present in the media. And also with the growth curve, there's a pretty standard logarithmic increase in cell concentration. And as expected, since there is no substrate, there is also no CLA production. However, when we look at the treatment group, upon LA exposure, the fermentation is actually slightly deviated with that of the control, specifically for JKL 2022, in which the fermentation is actually hindered. And in the growth curve, we can actually see that exposure to LA actually inhibited the growth of some of the strains, particularly JKL 2022 and 7019, in which we can see that they didn't reach 10 to the 9 and only reach a maximum of 10 to the 8, even after 24 hours of incubation. However, we can then see that the strains actually converted conjugated linoleic acid from linoleic acid substrate, and that the strong CLA producers are actually JKL 2022 and 7017. So in order to reduce the observed LA toxicity towards the bifidobacterial cells, we tried emulsifying linoleic acid in higher twin 80 emulsion. So that is, we used 2% and 5% of twin 80 and observed that at higher twin 80 concentration, there is better fermentation, higher cell viability, and higher CLA conversion for some of the strains. So that means that emulsification of linoleic acid in higher twin 80 concentration protected the cells from cell membrane damage and oxidative stress caused by linoleic acid. Moreover, we performed some optimization experiments for the CLA extraction method, in which we just tried to improve the efficiency of the existing isopropanol and hexane extraction method through the adjustment of the pH of the resulting mixture. And we can see in this figure that increasing the acidity to around pH 5 to 5.5 actually resulted in an increase in observable or measurable conjugated linoleic acid present in hexane. So this allows us to actually extract more of the conjugated linoleic acid from the in vitro reactions and get more accurate results. Also, we tried to determine if increasing the cell concentration would actually result in a proportional increase in the CLA conversion. And as we can see that with the increasing cell concentration and substrate concentration, there is a proportional increase in CLA conversion. And again, JCM strains using their washed cells or postbiotic preparations did not result in CLA conversion. Now, based on these experiments, we can say that the highest CLA yield from the 5x cell concentration and 10 mg per ml of free linoleic acid yielded approximately 8 mg per ml of conjugated linoleic acid. Now, this is comparable with the highest naturally occurring CLA concentration in food, which is around 7 to 8 mg per gram of fat. In addition to that, we also performed some gas chromatography or GCFID analysis in which we validated that the linoleic acid is preferentially converted into the cis-9 trans-11 isomer using the LAI enzyme of JKL 2022. So in another experiment, we actually tried to heterologously express the LAI in E. coli. Now, using the gene that we identified as LAI, we performed cloning experiments to heterologously express LAI and fusion LAI constructs in order to better understand the activity of this enzyme. So for that, we use the PGE-MTEC vector and the PET22B expression vector, as well as the PMAS C6T vector systems. And here's the overview of how I constructed the fusion LAI proteins that you can pause or screenshot and study at a later time. After several validation studies, we were now able to confirm that the linoleic acid isomerase is encoded by the gene JKL00014, which contains 993 base pairs. So this will have 330 residues with an isoelectric point of 8.48, a molecular weight of 37.2 kilodalton, an aliphatic index of 98.76, and an enzyme class of EC 5.2.1. We also determined the binding site, which includes the following residues and some of the basic characteristics of the enzyme. So we can say that linoleic acid isomerase is a membrane-spanning protein with 9 transmembrane domains that is highly conserved among Bifidobacterium breve strains. The conserved domain is also present among non-LAB GIT microbial population, and the most important thing is that it is different from the CLA-converting enzymes found in lactobacilli. Now on this side of the slide, we can see the predicted membrane topology of LAI in Bifidobacterium breve and the fusion LAI-SGFP in E. coli. So we can see that it is present as a transmembrane protein. So in order to verify this prediction, we actually expressed the LAI-SGFP in E. coli and observed the autofluorescence of the protein. So we can see in section B of this figure that there is strong autofluorescence signals coming from the periphery of the cells, whereas GFP-tagged proteins expressed as cytoplasmic membranes would actually give an equal autofluorescence signal all throughout the cells. One more particular observation that we saw in this experiment is that the fusion LAI proteins are actually much more upregulated or much more expressed when the inducer molecule is present at lower concentration, which is in contrast with most of the other overexpression studies. So we then validated the activity of the recombinant proteins using the same in vitro experiments, in which we first overexpressed the proteins in E. coli and harvested them through sonication and protein extraction methods. The membrane-spanning nature of the LAI posed as a significant challenge in the extraction of the proteins. However, the disruption of the cell membrane using a detergent such as Triton X100 improved the LA extraction and, in turn, the CLA conversion. However, we were only able to purify the LAI-SGFP fusion construct since the SGFP allowed the confirmation of the localization of the enzyme in the recombinant E. coli BL21. So you can see it in this figure. So the CLA converting activity of recombinant LAI is actually comparable with that of the native proteins found in Bifidobacterium breve. And from that, we were able to optimize the purification scheme and enzyme activity for the recombinant fusion LAI constructs. So using the recombinant E. coli, we designed a lysis buffer and an extraction buffer that will allow us to get 3 different cellular subfractions, namely the CFE or the cell-free extract, extract number 1, and pellet number 2, which we observed to have CLA converting activity. And in here, we can see that the relative CLA and CLNA conversion of the recombinant proteins are actually comparable with that of the native LAI proteins found in Bifidobacterium breve JKL 2022. And again, JCM7017 failed to produce significant amounts of conjugated linoleic acid when using its cell-free or membrane extracts. And lastly, we performed some biochemical assays wherein we compared the washed cells, the CFE containing the semi-purified LAI, and the purified recombinant LAI to determine the optimal reaction conditions, reaction temperature, and residual activity after temperature exposure. So here we can see that the CLA converting activity of JKL 2022 washed cells, semi-purified LAI, and the purified recombinant LAI-SGFP was evaluated under different reactions. So for the effect of pH, we see optimal CLA conversion within a narrow range of pH with high conversion rate, and that the optimal reaction pH is at around neutral pH or pH 7. As for the effect of reaction temperature, there is a wide range of temperature that showed high CLA conversion, but the optimal reaction temperature is around 30 to 40 degrees Celsius. And for the effect of temperature exposure, we can see that enzyme activity was lost after exposure to higher than 35 degrees Celsius, and high residual activity was only observed after exposure to temperatures less than 30 degrees Celsius. Now, from these results, we can say that the rapid rate of reaction from La into CLA allowed high CLA conversion at the following conditions: pH 6 to 8 and temperatures of 20 to 60 degrees. Knowing these biochemical parameters, it will be easier to proceed with the applications of the bifidobacterial LAI into several industrial applications considering these factors. As such, we will then proceed with the industrial potential of bifidobacterial linoleic acid isomerase. So one of the most direct application of bifidobacterial LAI is through the development of Bifidobacterium breve as probiotics. Now we ask the question, how do we develop probiotics? Starting with a strain of interest that shows the beneficial properties. Now in this case, it will be the Bifidobacterium breve JKL 2022 with strong CLA conversion. We can then proceed with several genomic and phenotypic characterization in order to prove the safety, functionality, and applicability of the strain. So we perform a simple experiment wherein we used a milk-based media to determine if the milk media can support the CLA conversion of Bifidobacterium breve. After several optimization of the milk media, either by adding glucose, yeast extract, or the combination of the two, we were able to observe good CLA conversion. So here we can see that RSM or reconstituted skim milk supported the growth and CLA conversion activity of JKL 2022 in both aerobic and anaerobic conditions, and that the supplementation of either 0.10% yeast extract or the combination of 2% glucose and 0.10% yeast extract yielded the highest CLA concentration. In addition, CLA conversion occurred only when there is curd formation, which is particularly beneficial for the production of yogurt and cheese. So based on these observations, we can deduce that even though the media and the bacteria was incubated under aerobic conditions, once there is curd formation, there will be a formation of a semi-anaerobic condition that would allow further growth and CLA conversion of Bifidobacterium breve, thus allowing CLA conversion in the anaerobic conditions. Another possible strategy for utilizing the bifidobacterial LAI is through the utilization of vegetable oils as a substrate for the in vitro CLA production. As mentioned in the introduction earlier, vegetable oils can be chemically processed to produce CLA supplements. However, they produce a mixture of CLA isomers, which is not optimal given that the beneficial effects of conjugated linoleic acid is actually attributed to the specific isomers. As such, we can leverage the isomer specificity of the LAI enzyme of JKL 2022 in order to produce high concentration of pure cis-9 trans 11 isomers that we can then use to fortify the conjugated linoleic acid content in dairy products through exogenous supplementation. Thus, we can enhance the CLA content in whole milk, cheese, butter, and other dairy products. Now, in order to determine the feasibility of this idea, we designed an in vitro digestion model for CLA conversion using vegetable oils as substrate. So we used perilla seed oil as a source of linolenic acid and grapeseed oil as a source of linoleic acid. We then determined the effect of cell concentration and reaction time, the effect of oil content, the effect of lipase concentration, and the total CFA production from either of the vegetable oils. And lo and behold, we observed high conjugated linoleic and linolenic acid production from either of the vegetable oils. So this is done through the combination of two different enzymes, which facilitates the two-step enzymatic reaction. First is lipase, which liberates the linoleic acid or linolenic acid from the glycerol backbone of the triglyceride in order for it to be available for the activity of the LAI enzyme. Now, although this approach is crude and still needs to be refined, it presents a strong evidence that vegetable oils can be used as a cheap source of conjugated linoleic acid utilizing the microbial enzyme systems for the isomer-specific CLA bioconversion, which can then be used as an alternative method for the chemical synthesis of vegetable oils for CLA production with higher isomer specificity. Now, these are just some of the other proof that the exogenous supplementation of conjugated linoleic acid or linolenic acid derived from the vegetable oils can be incorporated into dairy products. So CLA concentration in milk during the 2-week storage was stable during refrigerated conditions, reaching up to 396.47 increase in the total CLA content. However, for CLA concentration in yogurt, it was actually fluctuating, which is because of the low pH, which affects the fatty acid oxidation in yogurt. Therefore, we can say that milk is a better vehicle for CLA fortification. However, as mentioned, this approach is still developing, but there are already a lot of different studies directed towards enhancing the CLA content of various dairy products. Now, to summarize, the Bifidobacterium breve JKL 2022 strain demonstrated probiotic characteristics in vitro. This includes acid tolerance, bile salt resistance, and decent hydrophobicity with a wide range of fermentable carbohydrates. Now, although this was not mentioned in this presentation, most of these characteristics are actually innate for Bifidobacterium breve. In addition to that, JKL 2022 exhibited superior CLA converting activity, wherein it mediated the CLA production with growing cells or probiotic cells, washed or postbiotic cells, and crude protein extracts. In addition to that, the linoleic acid isomerase, which is a membrane-spanning enzyme, was successfully cloned and expressed in E. coli, and that LAI was characterized based on its activity and physicochemical properties. Lastly, Bifidobacterium breve efficiently converted conjugated linoleic acid from vegetable oils, which improved the CFA content in dairy products. Now, from all of this, we can say that Bifidobacterium breve JKL 2022 is a competent candidate as a probiotic or postbiotic preparation. So some of the applications include increasing the CLA bioavailability in animal-derived products through animal probiotics or postbiotics, CLA enhancing adjunct culture for various fermentation schemes using fatty acid-rich vegetable oils, as well as the development of a JKL 2022 functional food ingredient or an alternative therapeutics, which can be tailored probiotics, focusing on enhancing the CLA production to elicit immunomodulatory effects. And here is a graphical summary showing the overview of how we studied Bifidobacterium breve, focusing on its CLA conversion in order to produce CLA-enhanced dairy products. And that ends my presentation, and I would like to thank the New Agro-Food Research Institute of Chung-Ang University for the opportunity to share this study. I hope that you all learned and gained something valuable from this lecture. And if you have any questions, please feel free to contact me through my email. Thank you.